Welcome back to the playlist on heme synthesis. In this video, we're going to go over the physiology, biochemistry, and the organic mechanism of coproporphyrinogen oxidase. You can look at the systematic name, and you'll see it's coproporphyrinogen 3 oxygen oxidoreductase. And the reason it's called this is because it's going to use oxygen to perform an oxidation of this coproporphyrinogen. Okay? Now, I'm actually really excited to do this video because they've actually recently come out with a new proposed mechanism for this enzyme, and that's the one I'm going to do in this video. Okay? Before we really get into the details of this reaction, I have to kind of show you in general what we're looking at for the subcellular compartmentalization of this particular enzyme. So basically what we have is we have a cell. So here is our cell, okay? And of course, we have the mitochondria in here. Of course, I'm grossly over sizing the mitochondria. And then, of course, in the mitochondria, we have this inner membrane right here, right? And the space between the outer membrane and the inner membrane, what's that called? That's called the inner membrane space. And then, of course, in the center of the inner membrane, that region in there, that's the mitochondrial matrix, okay? So the thing you have to understand about heme synthesis is, remember when we did delta aminolevulinic acid synthase, we used ALA synthase in the mitochondria and we transported ALA out into the cytosol. So out here, this is the cytosol, and that's where ALA is going to go through various transformations. So we're going to start with aminolevulinate or delta aminolevulinate. And we're going to, of course, react that with a series of enzymes, starting with porphobilinogen synthase. So we're going to do a series of enzymes, and that's ultimately going to get us to something called coproporphyrinogen. I'll abbreviate that as CP. Okay. Now, coproporphyrinogen is going to finish the transformations into heme inside the mitochondria. Okay, so what has to happen is, through an unknown mechanism, coproporphyrinogen 3 has to be transported through the mitochondrial membrane into the inner membrane space. So actually the catalysis of this enzyme, coproporphyrinogen oxidase, has recently been found out to not occur in the matrix. This enzyme is purported to actually be soluble in the inner membrane space of the mitochondria. And the way that they figured this out was that if you preferentially destroy the outer membrane of the mitochondria, this particular enzyme sort of comes out, which sort of leads scientists to believe that it's not actually anchored in the inner membrane. It's actually floating free in the inner membrane space. So that's going to be the topic of this video, coproporphyrinogen oxidase. Okay, we'll get to the mechanism shortly and kind of analyze the structure of coproporphyrinogen 3. But first, let's talk about what happens if you have a deficiency of this enzyme. If you have a deficiency of this enzyme, what it usually means is you have a mutation, a genetic mutation in this gene right here, which is called CPOX. This is the gene that codes for coproporphyrinogen oxidase. And this mutation occurs in about 1 of every 20,000 people. And because it's a deficiency of an enzyme in heme synthesis, you term it a porphyria. So this is a porphyria. Basically, all the people who suffer from these porphyrias, it doesn't really matter what porphyria is, there's kind of a set of symptoms that these people suffer from. Those include things like nausea, vomiting, constipation, and then they also have skin problems. Basically, their skin becomes rather weak, and so it's prone to being burned by the sun. They get lesions, scars, all sorts of nasty things. That kind of goes with all of these porphyrias. But this particular one, hereditary coproporphyria, this is a deficiency of coproporphyrinogen 3 oxidase. Okay, so without boring you any further, let's actually look at the structure of coproporphyrinogen 3. Okay, what I want you to notice is several things. Number one, remember in the previous reaction, uroporphyrinogen 3 decarboxylase, all of the acetyl groups that were on these pyrrole rings, we basically decarboxylated those and we got methyl groups. So these guys right here, these are the methyl groups that were acetyl groups. Okay, And so what we're going to do in this reaction is we're going to decarboxylate, but we're actually going to do it with a rather different mechanism. Because in the previous mechanism that we saw for uroporphyrinogen decarboxylase, we weren't dealing with propionate groups. We were dealing with acetyl groups. So these groups right here, these things that we're going to decarboxylate, these are termed the propionate groups. These are the propionate groups. And it turns out that the ones right here shown on the top, these are the ones that are going to get decarboxylated. And essentially what's going to happen is they're going to be replaced with vinyl groups. So if you're curious what a vinyl group is, basically you have some molecule over here and then it looks like this. So this double bond right here that's now attached to the rest of the molecule, this is called a vinyl group. And essentially these propionate groups are going to be replaced with those. Okay? Now basically what we're going to do is we're going to use two molecules. We're going to use two molecules of molecular oxygen, okay? And what we're going to essentially get out is two molecules of hydrogen peroxide. So two molecules of hydrogen peroxide. So the oxygen in this reaction sequence is going to be what does the oxidation. In the process, molecular oxygen is going to get reduced ultimately to hydrogen peroxide, okay? And what we're going to get out of the end of this reaction is something called protoporphyrinogen. And specifically, it's protoporphyrinogen 9. Okay, that's what we're going to get at the very end of the sequence. I'll go ahead and show you the final product after the mechanism's complete. You can see here that these propionate groups have been replaced with vinyl groups. So that's basically what we're going to. Now we're going to actually look to see how that's done organically speaking. Okay. So basically the first step of this mechanism is going to be a proton transfer. So this pyrrole nitrogen, okay, we're going to transfer the proton onto a base in the active site. So I'll do the mechanistic steps in green. So first we're going to abstract this proton right here, and that's going to cause a double bond rearrangement of the pyrrole ring like this. And then these electrons right here, they will then go and attack molecular oxygen. And when that happens, one of the electrons, the radical electron that is going to be transferred onto the other atom of oxygen that was not attacked. And so what you ultimately get is sort of this, this negatively charged oxygen that's now free to do another proton transfer. Okay, so basically what we're going to do now is this oxygen right here now has a negative charge. Notice here that this atom of molecular oxygen right here, it was overall neutral. Right? It has basically 6 electrons to it, 6 valence electrons, and so that classifies oxygen as being neutral because it's in group 6. It's a chalcogen. Okay? But when we add this extra electron from the other oxygen atom, that have 7 electrons. So I could go ahead and draw the other electrons here. And what you'll see is that now oxygen has 7 valence electrons. And so it has a negative charge now. And now it can act as a Bronsted-Lowry base. And so what it's going to do is it's going to deprotonate this beta carbon right here. So if you're looking at the propionate group, basically the way you label these is the one that's closest to the carboxylate, that's the alpha carbon, and the one that is closest to the pyrrole ring, that's the beta carbon, or we could say it's most distal from the propionate group. And when this happens, okay, what you're going to get is another double bond rearrangement, okay? And so now this proton that was originally abstracted by the base is returned to the pyrrole ring. And so now we get this adduct right here between what was the pyrrole ring and what's now a peroxy species, okay? Now at this point, we're going to do the decarboxylation, and this is how it's going to occur. This oxygen is going to kick its lone pair in here, and at this point we're going to produce a carbon dioxide. So before the hydrogen peroxide comes off, I should denote that we're going to get a CO2. This is the first carbon dioxide that's produced. And then this bond, which I'm going to highlight in yellow, this bond right here is going to break. And when it breaks, these electrons are going to kick in here, and then you'll get another double bond rearrangement, but this time the double bond moves this direction and it kicks off a peroxide anion. Okay, and as soon as the peroxide anion picks up a proton from solution, you're going to end up with hydrogen peroxide. Okay, And essentially what's going to happen is hydrogen peroxide will react with catalase, and that's going to give you molecular oxygen and water. Okay, so the hydrogen peroxide has to be dealt with. It's dealt with by catalase. Okay, now this particular decarboxylation, this oxidative decarboxylation, in fact, let me write that down. This is an important term. This particular enzyme performs an oxidative decarboxylation, okay? And it's not just any decarboxylation, it's oxidative because it requires molecular oxygen. Some of the decarboxylations we've seen involve pyridoxal phosphate. That was the case in delta aminolevulinic acid synthase. Some of them don't require any oxygen at all. Okay, for example, a rhododilate decarboxylase, which is in pyrimidine biosynthesis. That is a very simple decarboxylation that doesn't require any cofactors or any other substrates. Okay, this one required oxygen, so it's oxidative. And I want you to notice something, that this part of coproporphyrinogen 3 that I circled in yellow, that's this ring right here. I'll label that ring as 1. Okay, so this pyrrole ring that we just decarboxylated, that's pyrrole ring 1. But now what we're going to do is we're going to do the same thing, but we're going to do it to pyrrole ring number 2, which is basically, if you're going around the porphyrin ring in this orientation, you're now moving clockwise. Okay, so now we're going to do the exact same thing to the second pyrrole ring. Okay, and notice once again, we got the vinyl group out of this. So now, again, I'll do the mechanistic steps in green. It's the exact same mechanism. So the base in the active site is going to deprotonate the pyrrole ring, and once again, we get a double bond rearrangement. And you guessed it, these pi electrons come out and attack molecular oxygen, and that forces this lone electron to move over to the other oxygen atom, generating a negatively charged oxygen that can now do a proton transfer. And just like in the case of the last one, here's your alpha carbon of the propionate group. This is, of course, your beta carbon right here, the one closest to the pyrrole ring and the one most distal from the carboxylate. Well, it turns out that that negatively charged oxygen will now deprotonate the beta carbon, and that's going to force another double bond rearrangement. And when it rearranges, these pi electrons will come out and abstract that proton from the base in the active site. Okay? And once again, now we're going to do the decarboxylation. Notice that we've essentially activated this carboxyl group for decarboxylation, because when we break this bond that I'm denoting in yellow, when we break this bond, we can rearrange the double bonds and get loss of a leaving group. Okay, so whenever you can do that, you essentially have an activated carboxyl group. So these electrons kick in here to form carbon dioxide. That's the very first thing that happens. So CO2 comes off, and then this bond breaks, kicking in here to form the vinyl group. This double bond rearranges over here, and it's going to kick off the peroxide species, or the peroxide anion, and when it picks up a proton from solution, it's going to become our friend hydrogen peroxide, or maybe not our friend, that's a toxic molecule. And then hydrogen peroxide will then react with catalase, and that gives us molecular oxygen and water. And so once again, you notice that we have the vinyl group now here on the second pyrrole ring of this tetrapyrrole. And now, after this transformation, we have this guy, which is protoporphyrinogen. Okay, and in the next reaction, protoporphyrinogen will react with protoporphyrinogen oxidase, and that's going to give us protoporphyrin. And then protoporphyrin will react with ferrochelatase to give us the heme B. Okay, so that's our final product. All of those transformations will now occur in the mitochondria. Okay, when we make the heme will then be transported out back into the cytosol of the cell. Okay, so now we've seen the mechanism of coproporphyrinogen oxidase, but what I want to talk about really quickly is two things. Number one, there is also a coproporphyrinogen dehydrogenase. This is a different enzyme. Okay, it does the exact same transformation, but it does it through a different mechanism. We'll probably have that eventually in a separate video, but what I want to get you to know is that this particular reaction, this is the one that's done in humans. Certain bacteria are able to do coproporphyrinogen dehydrogenase, And that particular mechanism does not use molecular oxygen. It uses S-adenylyl methionine, and it uses a mechanism called a radical SAM mechanism. We're not going to do that in this video. That'll be a separate video. But that particular enzyme, the radical SAM mechanism, as far as anyone knows, that's not a human enzyme. The one that occurs in humans is this one that uses molecular oxygen, Coproporphyrinogen Oxidase. And also remember this, think back to our other videos in the past, or maybe your prior knowledge. Whenever you hear the term oxidase, that should inform you that this particular enzyme is going to use molecular oxygen, but none of the atoms of those molecular oxygens get incorporated into the final product. So notice that in 2 of these steps, we used 2 molecules of molecular oxygen. Here's one of them right here. And then decarboxylating the second pyrrole ring, we of course use the second one. And we notice that none of those atoms end up getting incorporated into the final product, which is protoporphyrinogen. They instead get lost as hydrogen peroxide, and that gets catabolized by catalase. Okay, so therefore the term oxidase for this enzyme. Okay. There's one other thing I wanted to mention that's really impressive about this enzyme. In a way, it's sort of similar to rhododilate decarboxylase, which is that enzyme in pyrimidine synthesis that does decarboxylations without any coenzymes. Now, this is a very bizarre enzyme for the same reason. Okay, this particular decarboxylation, it does this without any coenzymes. Okay, there's no coenzymes known for this decarboxylation. Okay, in a sense, it's actually very similar to uroporphyrinogen decarboxylase. So I just find that really fascinating that you're able to do this reaction without any coenzymes. All you need is molecular oxygen, and certainly there's plenty of that around. It's really a fantastic reaction, and I really enjoyed doing this video because they actually just discovered this mechanism as a possible mechanism for the one that's done in humans. Okay, so I hope this video gave you a little bit of intuition on coproporphyrinogen 3 oxidase. In the next video, we're going to look at the transformation of protoporphyrinogen to protoporphyrin, which is done by protoporphyrinogen oxidase. See you in the next video.